This method can help answer key questions about perturbations in signaling networks that can cause cancer growth or the development of resistance to specific therapies. The main advantage of this technique is that it can provide rapid and reliable feedback on the phosphorylation state of multiple networks that are frequently deregulated in cancer.
The implication of this technique extends towards therapy of drug-resistant cancers because the identification of alterations in post-translational modifications can elucidate changes in signal transduction pathway activity. While this technique is used on phosphorylation, it can also be used to look at other post-translational modifications, such as glycosylation and ubiquination.
Begin by thoroughly rinsing overnight cultured cells 3 times with 10 milliliters of PBS, taking care to remove all of the PBS after the last wash. Next, add the appropriate volume of lysis buffer from the protein assay kit, and use a cell scraper to detach the cells. Triturate the resulting cell solution approximately 10 times before transferring at least 1 times 10 to the 7th cells to a 1.5 milliliter tube. Incubate the cells for 30 minutes at 4 degrees Celsius with shaking. Then, load the lysate into a syringe equipped with a 27-gauge needle and aspirate and dispense the sample 10 times to shear the cell membranes. When the cell membranes have been completely disrupted, centrifuge the lysate and transfer the supernatant to a new 1.5 milliliter tube for a subsequent total.